unlike other cells in the body that when they die, you replace them through replication, neurons in the brain you don't replace, or you replace to a very limited extent.
Welcome, everybody. This is Buck Joffrey with the Longevity Roadmap. And today I am doing a very interesting show with another member of the Buck Institute. You guys need to check out the Buck Institute. It is a crazy place where they're doing all sorts of interesting things. Of course, we've also talked to Dr. David Furman from there as well. This is a place where a lot of the future is happening in real time. Now, today we are going to talk to Dr. Julie Andersen. Dr. Andersen is interested in the brain and neurodegenerative disease, and how aging affects that. Fascinating thing. And one theme that continues to come up is the importance of mitochondrial health. And I am completely fascinated by this, especially with the concept of potentially infusing new mitochondria into our bodies. There is some research on that. There's also, obviously supplements and that kind of thing that we're going to learn a little bit about. Some of the challenges we have with these brain-related diseases and brain aging in general, though, is one of the things that keeps our brain safe, which is the blood-brain barrier. So Dr. Andersen is going to talk a little bit about that too. How do you overcome the blood-brain barrier? How do you deliver, the right drugs? And what else can we do right here and now in order to potentially slow down brain aging? And we will have that interview for you after these messages.
Hey, longevity enthusiasts, it's time to take it to the next level. I've been fine-tuning my longevity regimen for years, and I look better and feel better than I did a decade ago. In fact, my blood work is even better than it was back then. And it's all because of my data-driven regimen. And it's inspired me to create a course and community just for you. It's called the Longevity Roadmap, and I urge you to check it out. If you're tired of your belly fat, tired of being tired, or just want to optimize yourself for the next 50 years, visit longevityroadmap.com. That's longevityroadmap.com.
Welcome back to Longevity Roadmap. Today, my guest is Dr. Julie Andersen, a senior scientist at the Buck Institute for Research on Aging, where she leads a lab focused on the molecular mechanisms driving neurodegenerative diseases, particularly Parkinson's and Alzheimer's disease. She's earned her PhD in neurotoxicology and completed postdoctoral training at both Harvard and Massachusetts General Hospital. Over the past 2 decades, she has published extensively on the role of mitochondrial dysfunction, oxidative stress, glial activation, and cellular senescence in brain aging. Her research is helping to really redefine how we think about neurodegeneration, not just as an isolated disease, but as downstream manifestations of the aging process itself. Dr. Andersen, thank you so much for joining us.
Oh, thank you for having me.
Yeah. So why don't you just start off kind of, a little bit basic in that the brain seems to show signs of aging earlier and more dramatically than many other organs. What makes it so vulnerable to the aging process?
Yeah, if you think about the brain as being kind of the master regulator of the body, maintaining function is really important. One of the reasons it's probably a lot more vulnerable is you have cells called neurons, which are known to be post-mitotic, right? So they're cells which, unlike other cells in the body that when they die, you replace them through replication, neurons in the brain you don't replace, or you replace to a very limited extent. You have very limited number of neural stem cells in the brain. The brain also has really high, it uses a lot of oxygen, uses a lot of mitochondria, and which is wonderful because you're producing a lot of energy, but the byproduct of that energy production through this kind of powerhouse of the cell is that you have, as a toxic byproduct, oxidative stress or reactive oxygen species that can damage cells in the brain. So it's both very important organ and also very vulnerable organ to things that go on during aging.
Yeah. So, I've seen in your research, you really emphasize mitochondrial dysfunction as a key driver of, I guess, aging of the brain. So what are some of the most important upstream contributors to that dysfunction as we age?
I mean, honestly, the mitochondria, again, they're producing energy that allows all our cells to function. But because during the process of oxidative phosphorylation, you're also producing this oxidative stress, the mitochondria are also sort of producing damage to themselves. And that gets, it's sort of this idea that with aging, you have this chronic production of these oxidative byproducts in the mitochondria that can damage the mitochondria. So more damage. And unfortunately, as you get older, the normal repair mechanisms that either the antioxidants that get rid of the ROS that are within cells in the brain are reduced. And also the ability of the cells to get rid of the damaged mitochondria through a process called mitophagy, where the damaged mitochondria gets taken up into an organelle called the lysosome, which is kind of a little acid-filled sac
Mm-hmm.
within
Yeah.
within the cell, and they get broken down into components that are used to make new, healthy mitochondria.
Yeah. You know what? I want to get into some of the mitochondrial health targets, in a minute. But one of the things that I used to be in neurosurgery myself, and an ongoing theme, if that doesn't really apply to the rest of the body necessarily, is this blood-brain barrier. When you start talking about designing any kind of interventions for mitochondria, I guess the first question is, like, how far have we actually come in solving the delivery problem for aging-related brain therapeutics?
Yeah, that's a great question because as you're alluding to, you can give a drug which helps kind of juice up the level of antioxidants or the ability of cells to turn over damaged proteins and mitochondria to the lysosome. But in the brain, they have to get across this additional blood-brain barrier,
Yeah.
which is important because that sort of is what keeps toxins out of the
Yeah.
brain. But it is kind of an impediment. Um, I would say there are some exciting new technologies towards that, including use of kind of mechanowave systems, but there's also use of engineered, for example, one of the things my lab is interested in is using immune cells from the body and engineering them so that they basically hone in to the blood-brain barrier and then can be passed into the brain across that barrier. So these kind of more targeted therapeutics that are going to be selectively allow entry into the brain are important. Yeah.
I'm just curious specifically on that. I think you mentioned maybe some focused ultrasound type device. How does that work?
It's just kind of by temporarily disrupting. Other
Uh-huh.
people use altering the osmolarity of this system. So a lot of what has been looked at in the last couple decades is really sort of a means of temporarily, opening the blood-brain barrier. Although the problem is that if you're using a bulk therapeutic, like you're giving something IV and then you apply this technology, yes, the drug can more easily get across the blood-brain barrier, but also whatever else is there as well. So this kind
Yeah.
of idea of cell-based delivery
So it's
where
not terribly specific, which can be
right.
problematic. So something instead like, exosomes or, ligand targeted nanoparticles, things like that.
Right, exactly. And nanoparticles as well have, come to the fore in the last decade or so for use of something because the blood-brain barrier too, it's like endothelial cells, but their membranes are composed of lipids and they have kind of selective lipid compositions of those membranes that you could target. And also there are selective receptors that allow transport of agents across the brain, like transferrin for iron, for example.
Are any of these things that we're talking about right now actually being used in clinical practice, or are they all kind of, sort of in this theoretical laboratory phase?
My understanding is there are some of these technologies that are actually in phase 2 and phase 3 trials, generally for things that are kind of more acute as opposed to long-term treatments for, I mean, my laboratory studies diseases like Alzheimer's and Parkinson's disease.
Yeah.
So I think that there are trials that are ongoing. I have to admit I'm not an expert on those, but
Yeah,
they're out
sure.
there.
Understood. Okay, well, let's turn back to, again, we're focusing on mitochondria, and you hear about so many compounds that presumably in one way or another have some positive benefits for mitochondria. Which ones are you focusing on and why?
A lot of our work is focused on, I guess, what I would call lysosomal rejuvenators. So they're agents that are sort of increasing the capacity of the lysosome to get rid of damaged proteins and mitochondria. And this sort of came from a study that we published 10 years ago in J Neuroscience, where in looking at a Parkinson's mouse model that expresses a mutation called PARK2 or Parkin that you see in some familial patients with Parkinson's disease. If you express that human mutation, the mice actually, I mean, they're not a perfect model. No mouse model is a perfect model, but they do show hallmarks of Parkinson's disease, including specific loss of these dopaminergic midbrain neurons that are involved in regulating motor movement in the substantia nigra, and buildup of protein aggregates within cells called Lewy bodies after Dr. Lewy, who originally described the disease over 200 years
Sure.
ago. And we found that if we gave this compound called rapamycin, which your audience may have heard of, rapamycin, one of its known functions is to upregulate the master transcriptional regulator of lysosomal and autophagy genes called transcription factor EB or TFEB. And if we upregulated TFEB through rapamycin in these mice, it prevented, if we did it before they showed disease development, that it prevented both the neuropathology that you see, the alpha-synuclein Lewy body deposits, loss of the dopamine neurons, and also they showed improved motor function in these guys.
So it's interesting. It's actually a different mechanism than the usual mTOR pathways that we associate with rapamycin.
Yeah, it actually independently is through, or I should say that rapamycin by inhibiting mTOR blocks TFEB from going to the nucleus and turning on gene expression. But the agents that we look at are actually directly. So what happens with mTOR is mTOR actually usually phosphorylates TFEB, and that's what prevents it from being translocated to the nucleus and inducing gene expression. In our case, with the agents that we've identified, they specifically increase transcription of TFEB. So it upregulates gene expression of the master regulator. So yeah, it's slightly different. A lot of the agents that affect TFEB are through post-translational modification like you get with them.
Is rapamycin crossing the blood-brain barrier in this situation?
It does. How well it does, I'm not sure, and
Yeah,
that's
yeah.
always a question, right, with any of these agents.
And in these animals, when you do that, are these doses the types of doses that, in humans, people in the longevity space are pulsing, say, maybe 6 to 10 milligrams per week, maybe once per week? Is it that kind of thing, or is it like higher doses, or?
In the case of this paper from 10 years ago, it was more in the micromolar range. So, and it was giving for a certain period of time. So probably a bit different than what people are, currently using for dosages. And also, as you're aware, there's some immune side effects with rapamycin, which
Sure.
is why people have been so interested in looking at these rapalogs or drugs that are kind of look like rapamycin, but you're hoping you get reduced
Yeah.
toxicity or side effect. Yeah.
You've also looked at senolytics. These, just as a reminder to our audience, compounds that basically clear senescent cells are sort of these cells that kind of get trapped in this phase where they're neither really living nor really dying. How viable do you think those senolytics are for the brain, given sort of the challenge of cell specificity, brain penetration, the fact that I don't even know how frequent you get senescent cells in the brain? It's curious.
Yeah, those are all really good points. There there are concerns in the sense it's sort of the grandfather of senolytics, things like D and Q and Navitoclax. It turns out Navitoclax, there was a publication about 5, 6, 7 years ago from the Mayo Clinic where they were treating mice that were expressing genes for frontal temporal lobe dementia, and they were giving those mice Navitoclax, and it seemed to prevent. So with frontal temporal lobe dementia, you have buildup of another protein called tau in that disease, and they found in those animals that giving them Navitoclax, the senolytics, seemed to prevent the tau pathology, and they seemed to do better cognitively. But it turns out that Navitoclax doesn't really cross, or I think the general consensus is that it doesn't cross the blood-brain barrier very well. So it's not clear that whatever effects that it's having aren't secondary effects, something going on in the periphery that then is impacting on brain function. The other commonly used senolytic DQ is been in clinical trials for Alzheimer's disease, and there was a phase 1 study that came out, and basically in that study, and I can't remember exactly, I assume it's mass spec, but they were able to show that Q crosses the brain barrier, but not D very well. Or I could have that backwards, but so there was some worry about the senolytics. You have to get over, as we've been talking about, that barrier, blood-brain barrier. And then there is an issue with selectivity. We actually have a paper that's under review where we've taken human primary cells and cultured them out long term so that we have neurons, we have other glial cell types, other non-neuronal cell types within this culture, like astrocytes. And we tested both of those DQ and Navitoclax in these mixed human brain cultures and I will say that these cells in those cultures look like they're mature cells, mature neurons, mature astrocytic character, that if you could find a dosage that was selectively getting rid of, let's say, senescent astrocytes, it was killing normal neurons. So I think, again, having these directed therapies rather than giving a
Yeah.
whole
Yeah. Yeah.
drug, having a directed cell-based therapy, I think is going to be important, just generally, but also for senolytics
Yeah.
or xenomorphics.
So you stepping back to mechanisms in general. So you've talked about oxidative stress, the reactive oxidative species from the mitochondria themselves. I think you've written also about, protein misfolding in those aggregates and inflammation, and these things forming sort of this self-reinforcing triad to the aging brain where, it's getting worse and worse, like this
Yeah.
cycle. So if you look at that in the big picture, where do you see the best opportunity to break that cycle?
Yeah, I mean, honestly, because I think with a lot of therapeutics for neurodegeneration, I think we're giving them too late. So you're giving them after there's been a massive amount of cell damage and cell loss. But in order to give them earlier, it really requires, which is something a lot of people are working very hard on, is to identify early biomarkers of disease. I mean, theoretically, before you're showing memory loss or motor loss with Parkinson's disease, and there's some hints, for example, with like Parkinson's disease, we know Now, all the research was kind of on the specific subset of dopamine-containing neurons in the midbrain that are involved in motor movement and their loss. And then over the last decade or so, we've come to realize that it's kind of a systems disorder. Something like 80% of people who subsequently are diagnosed with Parkinson's disease have some kind of early gastrointestinal issues like IBS, or they have issues with smell, olfactory perception. And I think there's a lot of push towards looking at several of these together in terms of, and using now AI and machine learning with data from PD patients, like how much, whether we can figure out an algorithm that would say, okay, yes, you have X amount of chance, right, of developing Parkinson's disease later in life. So I think those kind of analyses have been a big boon to scientific research in
Mhm.
general, although the caution is make sure the data is good.
Right, right, right.
Uh,
Switching a little bit topic here wise. So there's this idea gaining traction that aging, systemic aging, aging in the body may actually start in the hypothalamus itself. Could you walk us through that model and whether you think it, I guess, whether, that probably applies to neurodegeneration as well, but just in general, like, what is the thought here?
yeah, I mean, I will say not my area of the
Yeah,
brain.
yeah.
But general, the hypothalamus is involved as kind of, I mean, I would say kind of the major sensing portion of the brain that's sort of sensing outside, like thirst or heat or, whatever outside environmental sort of things that are going on. It's, involved in light perception, etc. It's a very small area of the brain, but it clearly plays a huge role. And yes, it seems to, with aging, like a lot of other areas in the brain, you seem to have deficits. And frankly, I feel like it's been studied for as long as I've been around in terms of aging, but I feel like recently it's gotten a lot more, I'd say, deserved attention study of that
Yeah.
brain area, including at the Buck. There's a few labs here that are looking at
Yeah.
that.
Well, let's move back to what, you do, which is, a lot of work on Alzheimer's and Parkinson's at the basic science level. So these have, they affect, as you've talked about, they affect different parts of the brain. They involve different proteins. But what do you see as the most important mechanistic similarities between the two?
Yeah, this is what's kind of cool about this concept of geroscience. So geroscience is this idea that, and it seemed heretical 20 years ago, but it
Yeah.
was like not only is aging a major risk factor for diseases, and frankly, very disparate diseases, cardiovascular disease, diabetes, Alzheimer's. Not only is it a risk factor, but there are these underlying cell processes that can drive damage. And interestingly, a lot of those processes are shared across, different organs. Each cell and organ in the body has its own special characteristics, as we've sort of discussed with the brain. So I will put in a plug for the Buck and say that this term geroscience came out. We submitted through the NIH roadmap a center grant that we called geroscience, and it was exactly this idea that aging's not just a risk factor, but there's these common shared mechanisms. It got picked up by the NIA, and they started having geroscience symposia. There was a white paper that came out in Cell in 2014. Kennedy, that many people in the field were a part of, including people from the Buck, and it's just kind of exploded from there. And that 2014 Cell paper was really kind of espousing these, at that time, 7 hallmarks of aging. So it included things like cellular senescence, mitochondrial dysfunction, stem cell depletion. I should be able to name all 7, but I'm not sure I can. And
Yeah, sure.
now we're
Yeah,
up to
yeah.
12.
Yeah.
In the last couple years, there was sort of an update of that. And the interesting thing is that, like, none of those is an island unto itself. It's like
Sure.
if you have mitochondrial dysfunction, you have oxidative stress that can impact on stem cells, can impact on protein folding. So there's a lot of interaction between those hallmarks.
Aggregates, right? Like that's a big one.
Yeah, 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 for sure. And it's
Buildup of toxic protein aggregates, which is again what you've talked about before, is one of your targets.
right. Yeah.
So let me, I guess, one last question for you before we cut out here, which is, okay, we're talking about a lot of cutting edge therapeutics. Right now, what's the most evidence backed lifestyle intervention? Or you mentioned potentially, and I'm not asking for recommendation. We never recommend anything. But when you think about the things that we currently have access to, what do you believe? I mean, that actually makes a difference, whether it's lifestyle, supplements, drugs, anything like that, that's currently available to people.
Yeah, there's a lot of emphasis these days on supplements, but there's kind of this issue, and I think we're getting there, of like personalized medicine. So
Yeah.
supplements for one person may not be ideal for someone else, depending on their genetic makeup, which is different. I think we're kind of starting to understand what that means a little bit more. And I hate to be boring, but exercise.
Yeah. No, it's true. It's over and over again. That is the theme, especially
Yeah.
aerobic and resistance training, especially
Yeah,
things that we don't
especially
talk about.
when you're old, or strength training, because it turns out that you lose muscle cells with age, but you can increase the size of the cells through exercise. So there's compensation, right, through exercise. There are some things coming out that seem to act as mitochondrial mimetics by kind of juicing up the same things that get juiced up in young mitochondria, and replacing, mitochondrial infusion.
Yeah, what do you think of that? I've seen that around. Is that a
I think that that's a really interesting concept, in the sense that
Mhm.
you're giving the body, like, instead of giving some drug that maybe only impacting on one function of the mitochondria, you're actually giving mitochondria. I don't know enough about that to know, like,
Yeah.
I know it's gotten, there's a lot of excitement around it,
Yeah.
for
One of the
sure.
things I think is interesting that I learned, in doing a little research there is I didn't realize that mitochondria migrate.
Yeah.
Yeah.
It's funny because all our cartoons are sort of this round thing with
Yeah.
squiggles, and it turns out they're also like they don't look anything like the
Yeah.
cartoons we
Yeah.
draw.
And they
Yeah,
can
and
move
they
from
might.
one cell to another, right?
Yes, exactly. So there is a lot of, you know, cell-cell interaction, just like between the astrocytes and neurons. In terms of cellular senescence, the astrocytes, which are immune cells, if they become senescent, they can release inflammatory factors that can
Mhm.
affect neurons. So there are a lot of cell-cell interactions
Is
too.
there any I'm just keep very intrigued by this idea, but would there be any limitations to mitochondrial transfer with the blood-brain barrier?
I don't know. That's just what I was, it's
You were
probably
thinking the
easy
same.
to look up. I don't know.
Yeah.
Y I don't know.
Yeah. No, it's a fascinating idea because, I mean, if you could, and presumably, when they do that, I think some of it is about, like, harvesting your own platelets or whatever,
Right, exactly.
yeah, so it's not like it's foreign stuff, it's you're just
Right,
amplifying it.
exactly, and
Yeah.
that's kind of with using immune, like, TCARs or using these
Yeah.
immune cells. The idea is, even with things like glioma, that those cells can get into the brain. You would think, if a cell can get into the brain, mitochondria. I just don't know what the
Yeah.
strictures are in terms of how well and do they survive and are they functional, and I suppose there's other means of getting them in there.
Yeah.
Yeah,
Lots of stuff out there. There's probably somebody at the Buck looking into that, right?
yeah,
So,
for
yeah.
sure.
Well, thank you so much for your time. It's great to get your perspective on all of this stuff, and wish you the best on your research. And we'll certainly be following on the Buck Institute's website to look into
Yeah,
your
thank
research a little
you.
bit more.
Yeah, no, it's great. The Buck is really wonderful. It's a very interactive place. So, like we all are working together
Yeah,
towards
it's
trying
very
to
cool. Very cool.
figure out age-related disease. So,
Yeah.
great.
Thanks again.
Thank you. Thank you for having me.
Thanks for listening. A quick reminder that while I am in fact a surgeon, nothing I say should be construed as medical advice. Now make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe.